Buongiorno tutti, and welcome to the varvis workshop here at the SIGO conference, the virtual edition. This is our workshop on whole exome analysis and how it is ready to serve as a single assay approach for clinical genetics. My name is Yvonne Kassmann, and together with our two speakers, now I have the control, thank you. We will present today, the webinar is divided in two parts. The first one is on validated CNV analysis for WES and how it is enabling you to increase your diagnostic yield with confidence. And the second part is on the whole exome analysis and how why this is the method of choice when diagnosing intellectual disability. We will conclude with a question and answer session. However, we would like to not only have the interactive part at the end of the session, but to have the whole workshop in an interactive way. For this, we are decided a tool called Slido, and we are asking you all to actively participate in this in our Q&As and polls, live polls. We have distributed within this workshop. So in order to participate, this is very easy. All you need is your mobile device you have with you, and you open a browser on your mobile device and type in the address field, you type slide.do, and then you would enter the hashtag here. This is something that your monitor looks something like this. So you enter the hash here, which is varvis. We also have this in all the slides. You always see this, the hash, so you can always join on also later. And then you see two different tabs here, the Q&A tab and the polls tab. And so if you have questions to our speakers, you can type in your questions here, and we have a final slide where all the questions will appear. And we also have several polls that are activated in the slides, so where you can participate on your mobile, and you will show the results live in the slides. There's a short delay between what we present here and what you see in the webcast. So the polls will be active a few seconds before they appear on the screen. So you can always take a look on your mobile, and you will see them a bit earlier, and you can already participate. So to get started, we have a test poll here. Hopefully an interesting question for Italian people, especially regarding coffee. How many cups of coffee have you had this morning? So you take a look at your mobile, and in polls, you should see the questions popping up. And by clicking, you can actively participate. And we see the results coming in. So this is basically a result we were expecting. Not enough coffee. Oh, more people are joining in. That's great. So thank you. I see you figured out how to deal with this, and we have some more interesting questions coming up. And we get started with the content part now. So before we get started, let me briefly introduce to you who we are here at Limbus. We are a software development company located in the north of Germany, right at the Baltic Sea, as you see on this map. We are quite a young team, as you see on this picture here. Very friendly people. We are a medical device manufacturer, which means that we develop and operate our products according to international medical device standards. Our product varvis is cloud-based and designed to aid in the diagnosis of patients. I would also like to mention that this is a registered IVD medical device also in Italy. So one slide regarding varvis. It's a complete software solution for NGS. And it's a cloud-native CE-labeled medical device software, which has a big advantage. There's no investment necessary from your side. So the IT infrastructure we provide to you, and you can process. It doesn't matter how many samples you want us to process. We can do this very quickly for you. We can provide equal performance across the globe. We can provide also immediate quality control feedback to optimize your laboratory processes, which is especially important for the CNV analysis. It scales easily from small panels to WES and also to whole genome sequencing. And having an automated and validated CNV analysis also has enables you to have huge cost and time savings in the lab, as you already you just need to process like one laboratory process and no additional processes.